Hey there, science enthusiasts! Ever wonder how life on Earth began? Well, buckle up, because today I'm diving into some groundbreaking research that might just answer that mystery. Researchers have discovered a plausible geological setting that could have sparked life as we know it. Imagine this: volcanic porous rocks, water, biomolecules, and a gentle gas flow. Sounds simple, right? But here's the kicker. This setting could enable nucleic acids, the building blocks of life, to replicate all on their own. Let's break it down. In volcanic rocks, water containing biomolecules evaporates due to a gas flow. This creates convection currents, causing dissolved nucleic acids and salts to accumulate at the gas-water interface. This setup, believe it or not, promotes the separation and exponential replication of nucleic acids. In the lab, scientists recreated this environment on a microscale. They subjected a water sample to pure water influx and an airflow. What they found was astonishing. The nucleic acids accumulated at the gas-water interface, separating and replicating without any need for temperature changes, which usually degrade nucleic acids. According to lead author Philip Schwintek, they investigated a scenario where water movement through a rock pore was dried by gas percolating through to the surface, a very common setting on early volcanic islands. This created the dry conditions necessary for RNA synthesis. Using a model of the rock pore, the team observed an upward water flux evaporating at an intersection with a gas flux. This led to an accumulation of dissolved nucleic acids at the surface, while circular currents forced molecules back into the bulk. Schwintek noted that within just 5 minutes, DNA strands had accumulated threefold at the interface, and after an hour, there were 30 times more DNA strands. This environment seemed perfect for nucleic acids to replicate. However, replication isn't the whole story. Strand separation is also crucial. Usually this requires temperature changes, but here, changes in salt concentration did the trick. Senior author Dieter Braun explained that the circular fluid flow at the interface, combined with passive diffusion, drove strand separation by forcing nucleic acids through areas with different salt concentrations. Using FRET spectroscopy, they measured DNA strand separation. Initially, the FRET signal was high, indicating double-stranded DNA, but as the water flowed, the signal dropped, showing single-stranded DNA. This setting caused significant salt concentration changes, driving strand separation. In the bulk water, salt and nucleic acid concentrations remained low. To test if replication could really happen, the team added fluorescently labeled nucleic acids and an enzyme for DNA synthesis. Amazingly, after 2 hours the fluorescent signal increased, but when they stopped the gas and water influx, replication halted. Braun concluded that this simple geological setting could have triggered early life replication. A gas flowing over a rock pore filled with water, without temperature changes, creating salt fluctuations that support DNA replication. This discovery opens up a whole new range of potential environments for life's origins, not just on Earth but possibly on other planets too. Mind-blowing, right? If you enjoyed this deep dive into the origins of life, don't forget to hit like and subscribe for more fascinating science content. Until next time, stay curious.